Module 12, Chapter 17, Your Endocrine System. There are 4 different types of intercellular communication within the body. Gap junctions were introduced to you back in A&P 1 in the histology chapter. Gap junctions are formed when special channel proteins in the plasma membranes of 2 cells fuse together and form a water-filled tunnel connecting the cytoplasm of one cell directly to the cytoplasm of the next cell. This allows for you to distribute your effect very quickly because whatever you have in the cytoplasm of one is passed directly into the cytoplasm of the next. You can pass various chemical mediators through your gap junctions. You can pass nutrients, you can pass ions, and remember, the movement of ions is the movement of an electrical current. So this is an electrical signal passing from cell to cell. You can also pass other types of signaling chemicals from cell to cell. Paracrines are another mode of intercellular communication. Paracrines are referred to as local hormones. Now, these local hormones are going to be transmitted through diffusion within a tissue. So what happens is a paracrine is going to be released by one cell. It's going to spread by diffusion throughout the interstitial fluid of the tissue and impact the functions of other cells within that same tissue. Paracrines, on average, do not leave their tissues, so they produce a local effect on a tissue, not a systemic effect on the entire body. You then have hormones, which is the focus of this chapter. When you're focusing on hormones, the method of signal transmission is going to be through blood. And when you start thinking about using blood to transmit your signal, the distribution range gets larger. It means that you can produce a hormone in one area of the body, but yet affect a target, a cell sensitive to the hormone, very far from the site of release. Last semester, you also learned about neurotransmitters, and you learned that signals are transmitted across structures called synapses, where the neurotransmitter is released by a presynaptic structure, and then it's going to bind to and affect the function of a postsynaptic structure. The distribution of your effects with this method of communication is very restricted. You're only going to distribute your effect to the cell that is the receiving structure in the synapse, so that postsynaptic structure. Now, when you're talking about the endocrine system, you're talking about glands, tissues, and cells that secrete hormones. The field of endocrinology involves the diagnosis and treatment of endocrine system disorders. Now, you're going to hear the term endocrine gland frequently this particular set of lectures. And when you hear that term, you want to think in terms of things like the pituitary gland, the thyroid gland, your adrenal gland, and your pancreas. These are those nice multicellular glands producing hormones. However, there are other organs and tissues that produce hormones. For example, you learned about the atria of your heart at the beginning of the semester, and your atria produce a hormone called atrial natriuretic peptide. Your adipose tissue, which most of you just refer to as fat, produces a hormone called leptin. So when you start dealing with the endocrine system, you are looking at endocrine glands, but you also have to look a little more globally to anything that produces a hormone. Now, you're also going to hear me using terms like target organ and target cell. Whenever you hear these terms, I'm referring to structures whose functions are altered in response to a hormone. Now, your homeostasis is regulated by 2 body systems. In A&P 1, we focused in on the nervous system and its role in regulating homeostasis. For example, you learned that the nervous system communicates using neurotransmitters and action potentials. You learned that this communication is conveyed from one cell to the next at synapses with target cells. You learned that the nervous system produces local, very specific effects because your neuron is in direct contact with that target. You learn the nervous system acts very quickly. It can act within milliseconds of being activated. It also can stop quickly once your stimulus ends. Now, the nervous system does adapt very quickly to stress, meaning that your nervous system is going to be great for handling short-term stresses, things that don't last that long. However, some of your stresses do last longer than just a few minutes to a couple of hours, which would be short term. Sometimes stresses can last days to weeks to months, and your nervous system will adapt to those longer term stresses, meaning it can't handle homeostasis regulation in the long term. So try to think of the nervous system as a crisis management system, managing the crisis. Now, the targets of your nervous system are typically your muscles, certain glands, your adipose tissue, and other neurons. The endocrine system, however, has the same goal. It has the same goal in respect of regulating homeostasis to maintain your healthy normal. However, it does it very differently. For example, your endocrine system communicates using hormones that are released into your blood, which allows for the hormones to act on targets that are very far from the site of release. Because we're using blood and blood flows everywhere, your hormones are going to have very general, widespread effects instead of very local, specific ones. Because it can take a little bit of time to build up the level of a hormone to the point where you can see a significant effect, it can sometimes take hormones seconds to even days to have a significant effect on the body. Now, this means it's going to take a little bit for the endocrine system to get going with managing your stress. And if it takes a while to kick in, it's also going to take a while for it to slow down, for it to stop. So your endocrine system continues to respond for a significant period of time after the stimulus actually stops. Now, the good thing about the endocrine system is that it adapts rather slowly. So if you have long-term stress, the endocrine system will continue to manage your stress instead of adapting quickly like the nervous system. So think of the endocrine system as being that long-term stress management. Now, with regards to targets, your endocrine system can basically target any cell in the entire body because blood flow, for the most part, is everywhere throughout the body. Now, when you start looking at your hormones, they come in three major chemical classes. Now, you will need to be able to place each hormone that we cover in this chapter into its correct chemical class. Table 17.6, which is what we have on the next two PowerPoint slides, will help you to accomplish this task. I would also recommend that you focus in perhaps on learning the hormones that are in the smaller chemical classes, and then just know if I give you a hormone not in one of those two categories, that it automatically has to land in the other. For example, you have steroid hormones and steroid derivatives. Now, what makes the steroid hormones related is that they're all made from cholesterol. And your steroid hormones include aldosterone, your androgens. The most abundant androgen in the male body is testosterone, calcitriol, which is your active form of vitamin D, corticosterone, cortisol, your estrogens and your progesterone, the estrogen and progesterones being your main female sex hormones. You then have a class called the monoamines. These are also sometimes referred to as biogenic amines. Now, the monoamines are very different from the steroid hormones. Remember, steroids are made from cholesterol. The monoamines are actually produced by modifying amino acids. For example, the amino acid tyrosine can be modified to form thyroid hormone, norepinephrine, epinephrine, and dopamine. The amino acid tryptophan can then be used to produce melatonin. Now, you might have heard an old wives' tale about eating turkey and getting tired. Back in A&P 1, I know that I cover melatonin when we talk about the pineal gland in the brain. And I mentioned that melatonin is sort of like your body's natural sleep aid. It makes you feel drowsy and gives you that desire to actually begin your sleep cycle. But that the pineal gland is activated in the evening when sunlight is getting lower. Um, so you have higher production levels in darkness. And there was the whole school of thought, the whole argument, that on Thanksgiving, people eat all of this turkey. Turkey is very rich in tryptophan, and that the reason that someone gets so tired after eating Thanksgiving dinner is that the tryptophan leads to the production of more melatonin, and the melatonin makes you sleepy. However, you have to think about when Thanksgiving dinner is usually given. It's usually given during the daytime hours. You know, some people eat Thanksgiving dinner at lunch, some more at 3 to 4 o'clock, but it's well lit. You're not producing melatonin at that time. So it's impossible with bright sunlight for the tryptophan from your turkey to make you tired right after the meal. Most likely what is happening is that you are pulling blood from other vital organs like the brain and from your muscles and diverting more blood flow to your digestive system to handle the breakdown of the really huge meal that you just ate. And that decrease in blood flow to the brain decreases oxygen delivery to the brain, less nutrient delivery to the brain. And it might make you a little tired because you're prioritizing your digestive system over everything else. Your last group of peptide hormones are called your last chemical class are called your peptide hormones. Now, looking at this slide compared to the one that you just saw, can you see why I recommended that you learn what hormones are in the smaller classes, and that if it's not a steroid and it's not a monoamine, that it would land here? Now, peptide hormones are chains of amino acids. Now, the oligopeptides, which are roughly 3 to 10 amino acids long, include things like angiotensin II, an antidiuretic hormone. You then have polypeptides, which are more than 10 amino acids in size, such as calcitonin and glucagon and prolactin. You also have glycoproteins. Remember, glycoproteins are composite structures. The main component of the chemical is a protein, but it has a carbohydrate attached to it. So this is a protein with a carbohydrate addition. And you'll find things like erythropoietin and luteinizing hormone in that particular category. Now that you've been introduced to the different chemical classes of hormones, let's talk about how they're produced and distributed. Now, some of your hormones are hydrophilic and some of your hormones are hydrophobic. And you have to remember that hormones are being transported in your blood, specifically your plasma. And you learned in A&P 1 that hydrophilic means water-loving, meaning these things are going to want to dissolve with water, meaning easy plasma transport. Others are hydrophobic, Hydrophobic means water-fearing, and these chemicals tend not to want to mix with water, meaning they're going to need to have a little bit of assistance in order to be transported in that liquid medium. But let's start with hydrophilic first. Hydrophilic hormones are free in your plasma, so they very freely circulate in the blood. Their effects are generally fairly short-lived because they have the ability to easily diffuse out of your blood and bind to receptors in your tissue. They're also tend to be broken down by enzymes, either in your interstitial fluid, your plasma, your liver, your kidney. These two alone usually cause these hormones to be removed from your blood completely in just a few minutes of time. In addition, these hormones tend to be a little bit smaller, meaning they're very easily filtered out by the kidney. So you can get rid of them in your urine as well. Now, hydrophobic hormones are going to need assistance. In fact, they're going to bind to transport proteins in order to become soluble in your blood. Back in the blood chapter, you learned about plasma proteins. And if you think about the albumins and the globulins, they performed a good deal of transport functions of lipids and hydrophobic chemicals. So we're going to use those plasma proteins to bind and carry our hydrophobic hormones. So if you hear the term bound hormone, it means that your hormone is physically attached to the transport protein. If you hear unbound hormone, this means your hormone is unattached. And it is these unattached hydrophobic hormones that are the only ones that have the ability to leave your blood and enter tissue. Okay? Remember, your plasma proteins do not normally leave the blood. So if your hormone is attached to a transport protein, it can't leave the blood. So you have to unbind the hormone in order to get it to enter tissue. Now, there are some benefits to having this transportation method. The transport protein binding actually allows for a longer hormone half-life. A half-life is the time it takes for the concentration of your hormone to drop by half within the blood. So your transport proteins basically allow your hormone to last a lot longer in your blood. Part of this is because the binding to the plasma protein helps to protect the hormone from enzymes in your plasma, liver, and kidney. Also, binding that hormone to that big clunky protein creates a massively huge complex. And that complex is going to be so large that these hormones are not going to be able to be filtered out at the kidney and placed into your urine. Now, these bound hormones are going to act like a hormone reserve. Think of the hormone reserve as a backup supply of unused hormones. So these are hormones attached to the plasma proteins. They're not being used to affect the body. They're just floating around in there, just sort of waiting in the wings. And we're going to use this hormone reserve to replenish what is called your free fraction. The free fraction consists of all of the unbound hydrophobic hormones. Now, normally, the free fraction ranges between 0.1 to 10% of the lipid-soluble hormones. Now, exactly what percentage a specific hormone is, that's going to vary, but the general range is only 0.1 to 10%. And your free fraction must be kept in homeostasis so that you have enough of these hormones available to actually leave your blood, enter tissue, and regulate homeostasis. Now, generally what will happen, and very simplistically what would happen, is if you have one hydrophobic hormone leave your free fraction and enter your tissue, your hormone reserve will release one hormone, add it to your plasma to replace the hormone that left. So for every hormone that leaves the free fraction, we're going to remove a hormone from the reserve in order to maintain our 0.1 to 10%. Okay, so now we know that hormones come in different chemical classes. You've heard that some are hydrophilic, some are hydrophobic, meaning they're going to have to be transported in the plasma differently. So now that we have gotten our hormones transported to a destination, let's talk about how they act on your cell. So let's talk a little bit about your hormone receptors and your different modes of action. Now, one thing you have to keep in mind is that the cells of your body have receptors for many different types of hormones. Now, when you hear that word receptor, there are some things you need to remember from A&P1. Receptor proteins have specificity, meaning the receptor for one hormone will not bind to any other type of hormone. So it's basically one receptor type binds one particular chemical. Your receptors can also exhibit saturation. You have a set number of hormone receptors at any point in time in the plasma membrane for a hormone. And if all of the receptors are occupied by hormone, there's no possible way to bind more and bring that hormone into your cell. You also have to keep in mind that your cells can have different combinations of receptors. So not every hormone is going to affect every type of cell in the body. So for example, you could have a cell that has receptors for testosterone, insulin, and thyroid hormone. You could compare it to a cell in a tissue in a different part of the body, except that cell has receptors for thyroid hormone, for insulin, and for melatonin, but it doesn't have receptors for testosterone. So not only can one cell have receptors for many different types of hormones, but from cell to cell, they have different combinations of receptors. Also, to boot, to make it even more complicated, to add another layer, in order to be stimulated by a hormone, a cell must have a receptor for that hormone. So if you don't have the receptor for it, you could be bathed in the highest concentrations possible of the hormone and the cell won't be affected. So no receptor equals no effect. So when you start thinking about going into a career like endocrinology, it is a very layered, complicated field because you have to account for all of the different hormones that are impacting your cells all at the same time. Now, when you start thinking about the receptors, they can be found in one of two places. Some of your receptors are referred to as plasma membrane receptors. So they're going to be positioned in the plasma membrane, sticking out into your interstitial fluid, waiting for hormones to come around that they can bind to. And some of your receptors are going to be tucked away inside the cell itself. So let's talk about the plasma membrane receptors first. Plasma membrane receptors are generally used for hydrophilic hormones that don't have the ability to pass directly through the plasma membrane. So remember, the phospholipid bilayer has a core that's very hydrophobic. It's made from those fatty acid tails. So in order to pass across the plasma membrane, you have to be just like those lipids. And your hydrophilic hormones love water. They don't love lipids. So they're not going to be able to come in. Now, the hormones that would use plasma membrane receptors are your peptide hormones and your catecholamines, things like epinephrine, norepinephrine, and dopamine. Now, how your mechanism of action will work is that you first have to form what is called a first messenger. The first messenger is the hormone physically bound to the receptor. So think of this as you have dropped off a letter at the post office. Okay, so you've delivered the mail. The only thing is that once your letter has been delivered to the post office, doesn't it have to then be taken to where you want it to go? You need someone to take the letter from the post office, carry it out into the great wide world, and actually transport that letter, that message, to where it needs to go. So it's sort of the same thing here. We have this first messenger, we have a hormone physically attached to the outer surface of your cell. Somehow, that hormone on the outside of your cell has to create an effect on the inside of your cell, which means once we have our first messenger, we have to form the mailman, the second messenger, who's actually going to carry our message inside the cell and lead to an effect, some kind of a change. Now, there are different types of second messenger systems. The most common is called cyclic AMP, or lowercase c, capital A, capital M, capital P. You can see cyclic AMP, that camp, up in the upper right-hand corner of the image. There's also something called DAG, which is diacylglycerol. Now, once you have cyclic AMP and DAG, they initiate a series of chemical reactions that leads to the phosphorylation of existing enzymes. Now, what that means is you're going to add a phosphate group to an enzyme you already have. And this phosphorylation could turn an enzyme on, or it could turn an enzyme off. Turning an enzyme on or off is going to alter the function of your cell, which is the point of hormonal signaling. Another type of second messenger that you could use is called IP3. It stands for inositol triphosphate. IP3's action is through initiating a series of chemical reactions that leads to an increase in calcium concentrations within the cell, which in turn leads to other physiological changes. Now, the use of plasma membrane receptors allows for something called signal amplification. Signal amplification allows one hormone molecule to actually trigger the synthesis of an enormous number of enzymes. So, for example, you get this little teeny tiny stimulus, so a small amount of hormone. Except when those hormones bind to the cell, they create a relatively large number of cyclic AMP, DAG, IP3. These chemicals then spread out within the cell and lead to an even higher number of activated enzymes. And you guys learned in AMP1, enzymes don't get used up. All they do, as long as there is substrate, is catalyze chemical reactions, make them happen faster. So once you have a pool of activated enzymes, you have the ability to create an enormous amount of product, which gives you a greater effect due to one little teeny tiny hormone hitting your cell. Now, the number of receptors in your plasma membrane can change to adjust your cell sensitivity. So, for example, there is a process called downregulation. Downregulation is going to decrease the number of plasma membrane receptors, which is going to make your cell less sensitive to a hormone. Well, when would you want this particular event to happen? Well, think about this. Imagine that you're a little teeny tiny cell in a tissue somewhere, and a hormone that you are sensitive to arrives, and it arrives in really high levels. So you've got this little teeny tiny cell. It is surrounded by an excessive amount of hormone. Well, normally, the more hormone you have, the greater the effect you will have on your cell. However, if you have abnormally high levels of your hormone, you don't want to have abnormally high levels of effect within that cell because it could push your cell out of homeostasis and potentially cause it to die. So, in order to prevent overstimulation of your cell, you will actually decrease the numbers of hormone receptors. Remember, I mentioned earlier with regards to your receptors is that they exhibit saturation. Once all the receptors are occupied by hormone, you can't bind more. So by decreasing our receptor number, we decrease the amount of hormone that we can actually bind, which helps to prevent overstimulation. Likewise, you can also perform upregulation. In upregulation, you're going to increase the number of plasma membrane receptors. By having more receptors, you increase the cell's sensitivity to a hormone. Now, this is going to be useful if your hormone levels are deficient. So you have an abnormally low concentration of hormone. But your poor little cell needs to bind a certain amount of that hormone in order to maintain homeostasis. So what will happen is if your cell is being understimulated, your receptor numbers will increase. And what it does is it's going to increase your cell's ability to find hormone in the environment and bind enough in order to keep your cell at homeostasis. Now, some hormones do use intracellular receptors. Intra means within. So these are receptors that are actually found inside the cell itself. These receptors are normally located within your mitochondria and within your nucleus. So in order to use intracellular receptors, the hormones have to be able to pass across the plasma membrane without any assistance needed. So we're looking at the hormones that are hydrophobic. So these would be your steroid hormones and also your thyroid hormones. So these hormones will diffuse across your plasma membrane. They will bind to receptors in your mitochondria or nucleus and have some very interesting effects on your cell. If the hormones are going into the nucleus, remember this is where your DNA is located. So these hormones have the ability to activate or deactivate specific genes. Remember, your DNA codes for all the proteins in the human body, and enzymes are proteins. So what these hormones can do is they can activate a gene to make a hormone from scratch, or deactivate a gene and stop it from producing a hormone. That's huge. You're affecting how many potential enzymes you might have to carry out your chemical reactions. When the hormones go into the mitochondria, they have the ability to actually increase the rate of mitochondrial activity, meaning your mitochondria are stimulated to produce ATP at much higher rates. Now, it can take hours to days for these particular hormones to actually show an effect on the cell. And this delay in seeing an effect is due to the lag time required for you to do things like genetic transcription, genetic translation, and just to accumulate enough of your protein product to significantly alter cell function.